Hey guys, hello, welcome all of you to Unacademy Future Doctors. This is Dr. Shivangi Shukla, and I am your educator for biochemistry. Do let me know if the live stream has started and if I am visible and audible to all of you. Let me just check once. Welcome all of you to Unacademy Future Doctors. Great. Okay, so we have started with metabolism in our focus session. Okay, this session is called Focus Prof 1. Okay, all of you who have been with me since the beginning would be knowing that we have come. This is a new This is a new channel on Unacademy. This is called Unacademy Future Doctors, and this channel is specially dedicated to MBBS Prof 1 and Prof 2 students. Okay, we have completed a super fast crash course in the earlier month, and we have also done a rapid revision series. Okay, now what we are starting with is the focus session. Okay, focus Prof 1 and Prof 2. These sessions aim at elaborate understanding and comprehension of the difficult topics. Okay, so I have started with the most often asked and the topic which maximum people have doubts in, that is metabolism of biomolecules. And yesterday we finished protein, 2 classes on protein and 1 class on carbohydrates, and tonight we are having similarly 2 classes on proteins and 1 on carbohydrates. Okay, so this is the third in the series, third class on proteins. Okay, following all of you? Okay, so let me just quickly skim through the different types of subscriptions available on Unacademy for NEET PG, Iconic as well as Plus. Okay, the Plus subscription comes with access to video lectures from top educators across the entire country. You get to watch their live as well as recorded sessions. You get to attempt the live quizzes that they conduct, and you will also have access to their printed notes in the PDF format, so that you just have to focus on understanding. Okay, like these sessions which I am taking, these sessions on YouTube. Similarly, there will be sessions on Unacademy under the Plus subscription. Okay, and the most important thing, the USP of this subscription is that you can access these classes while on the go on any device of your choice. Okay, another type of subscription is the Iconic subscription, which comes with access to resources from Prep Ladder as well. Okay, do not forget to put in the educator code DRSHUKLA, Dr. Shukla. This code is going to fetch you an additional 10% discount. But if you use this code before January 5th, that is today or tomorrow, if you use this code for subscription, it will fetch you 22% discount. Okay, which is a huge amount, isn't it? The price is already very reduced. Okay, for 24 months, the Plus subscription comes at just 36,000. And if you apply Dr. Shukla, DRSHUKLA, it will fetch you an additional 22% discount on this. Okay, so if you're planning on subscribing, do subscribe to this platform, Unacademy, within these 2 days, January 4th and January 5th, for availing additional 22% discount. Okay, which is a huge amount. Now, we had started with proteins yesterday. Okay, we had started with the structure. What we have discussed was the structure and the properties of amino acids. Okay, right? All of you would have must be following all this that we have discussed yesterday. All right. Different types of amino acids, their properties, also their synthesis we have discussed. Okay, superficially only we have discussed the synthesis. We'll come back to it later on when we start with metabolism. Right now we are just discussing about the structure. Now, one very important topic is the structure of proteins, especially the different types of secondary structures. Okay, the primary, tertiary, quaternary structure, you just need to know about the definition and the different bonds that stabilize these structures. Okay, but for secondary structure, you need to know, and you need to know how to draw the structure in detail. Okay, very important. You'll always get some or the other short note and some or the other MCQ from this. Okay, extremely important. Also, you should know the exceptions, the types of angles, phi and psi angle, the types of bonds involved, why right-handed helix and why anti-parallel beta-pleated structure is preferred in nature. All these things very important, you should know. Okay, so first of all, we should know the difference between these two terms that we are going to come across a lot. Hi, med student Jatin. Med student is saying biochem is so volatile. Revising what I read daily here. Prof exam on 29th. Your prof exam is on the 29th of January, is it? Then I would advise you to go through the super fast series. Okay, because there we have discussed all the important topics. Okay, please go through the super fast series. You can play the videos in on 2x speed and go through them, and also attend also watch the videos on Unacademy that I have taken. Okay, try and watch as many videos as you can on 2x speed, that is twice the speed, so that you don't waste a lot of time and maximum part is covered. Okay, through that you will be able to revise the maximum. Okay, Jatin is saying his prof is on the 14th. Okay, then you also need to revise. Okay, revising through video lectures takes uh, provides a lot of efficiency, because in less amount of time you're going to revise a lot. Okay, because you're going to just play the videos on 2x speed. Plus, also the audio visual learning is more consolidated inside our memory. So it is good that you can supplement the visual learning with the audio visual learning. Okay. Alright. So now this Jatin is saying a playlist like carb separate, protein separate. Jatin, you can play, you can use those, you can play those videos separately, right? Because everything has been marked, isn't it? So you can play them separately. Okay. Yeah, sure. I'll make a lecture on clinical biochemistry. Sure. You tell me which topics you need. I'll make a lecture on clinical applications. You need a lecture on practical biochemistry as well. I'll take that one as well for those of you who are going to appear in your professional examinations. Okay. Now, coming back to this, what is the difference between conformation and configuration? Okay, what is, now those of you who are appearing for your professional examinations, attempt this. What is the difference between conformation and configuration? Conformation is the 3D structure. Okay, how the structure of protein is appearing in 3D, that is the 3D arrangement. Okay, so it is the spatial arrangement of different atoms in a molecule. That is conformation. Okay, it is a higher grade of structure. And what is configuration? It is the geometric relationship between different atoms. Okay, configuration is the geometric relationship between different atoms, like, for example, the L and D configuration that we did yesterday. Okay, and the interconversion between two configurations requires breakage of covalent bonds, whereas interconversion between two conformations will not require any breakage of covalent bonds. Okay, it will just be different packaging. Okay, got it. Next, the 4 orders of protein structure. Most of you would be knowing: primary, secondary, tertiary, and quaternary. Okay, now as you go down, the folding increases, isn't it? The structure becomes more and more complicated because it becomes more and more folded. And what is the purpose of increasing orders of protein structure? It is to provide functionality to the protein, isn't it? It provides functionality to the protein. All right. Now, what is the primary structure? The aim of the primary structure, the primary structure is the sequence of amino acids in the polypeptide chain. Okay, it is just the sequence of amino acids in the polypeptide chain. It is like glycine, phenylalanine, methionine, glycine, phenylalanine, valine, cysteine. So this sequence of amino acids, this is called the primary structure. Okay, very basic. Alright, it is like the ID card, it is like the identity card. Now this chain, this primary structure, this is vanilla. Okay, now this protein wants to be very complicated for it to be functional. So what will it do? It will turn towards the secondary structure. Okay, this secondary structure is achieved when this plain vanilla primary structure gets folded. It can get folded into an alpha helix or into a beta pleated structure. Other secondary structures are loops, turns, bends. Okay, but they are not regular structures. The two regular structures are alpha helix and beta pleated structure. Okay, following, the third one is the tertiary structure. What is the tertiary structure? It is further up. When the secondary structure undergoes further folding, there are even more bonds that are formed. Okay, got it? And what is formed is domain. Okay, domains are formed. And these are the functional units. Okay, the functional proteins that exist, they either exist in the tertiary form or in the quaternary form. Okay, the quaternary form is even further folding to form oligomeric proteins. What are oligomeric proteins? That contain one or two different chains like monomers. What is a monomeric protein? Example, myoglobin. What is a tetrameric protein? Tetrameric protein is hemoglobin. Okay, so when different chains, different tertiary chains interact with each other, they form the quaternary structure. Okay, now four tertiary strains are interacting with each other, alpha 2, beta 2, to form the tetrameric quaternary structure of hemoglobin. Got it? Okay, understood? Now we'll start with the primary structure. Okay. Let me just check if you guys are following. Yeah. So, what is the primary structure? The primary structure consists of the peptide bonds. Okay. The peptide bonds are the main bonds present in the primary structure. What is the primary structure? It is the sequence of amino acid in the polypeptide chain. Okay. Now, what are the main, we have to read the main bonds that are present in each structure. What are the main bonds present in the primary structure? They are the peptide bonds. Okay, and what is one special characteristic about peptide bonds? That they contain partial double bond character. So, movement about the peptide bond is restricted because double bond is stronger. Okay. They have a partial double bond character, so movement about the peptide bond is restricted. Alright, this peptide bond is formed during the process of translation. We'll read about it. By which enzyme? Peptidyl transferase. Peptidyl transferase is what kind of an enzyme? It is a ribozyme. What is a ribozyme? Ribozyme is an RNA molecule behaving as an enzyme. Okay, so all of you, I jump from one topic to the other to revise. You guys should also do that while revising. Okay. Now, during the process of denaturation, what is denaturation? When you heat the protein or when you treat it chemically or through any other process, the structure, the conformation, the 3D structure of the protein is lost. The functionality is lost. Okay. And what remains is the primary structure. Why? Because the amino acids, they just remain holding each other's hands. So after denaturation, what remains of a protein is the primary structure. Okay. And this does not have any function because they are not arranged in a specific conformation. Okay. Understood the basic intricacies of the primary structure? Alright, do let me know if you guys are following. Now, the secondary structure, for before starting with the two important regular secondary structures, that are the alpha helix and the beta pleated sheet, we should know about a few important terminologies, angles that are going to be mentioned here. Okay, first is the phi angle. Phi angle is between the alpha carbon. It is the angle between the alpha carbon and the nitrogen bond. Okay, and the angle between the alpha carbon and the carbonyl carbon bond is the psi angle. How will you remember this? Psi, the syllable is C, right? This syllable is like C. So this will be between C and C. And this one is phi. So this is between C and N. Okay. Remember like this. Psi sounds like C. The syllable is C. So this will be between C and C. The psi angle is between alpha carbon and carbonyl carbon. And phi angle is between alpha carbon and nitrogen. Okay, so the bond that this the angle that this bond makes is called the phi angle. The angle that this bond makes is called the psi angle. Now these are the two angles about which there is free rotation possible in the protein. Okay. Why? Because the third bond, the third kind of bond, that is between carbonyl carbon and nitrogen, this is the third kind of bond, has a partial double bond character. Why? Because this is what is the peptide bond. Isn't it? Isn't it? This is, suppose this one is C, this one is NH2, this one is COOH, and this one is R. Okay. Now the angle is called what? This is the phi. Form a bond with another amino acid with the amide or with the carbonyl carbon amino acid. This bond is, what is this bond? This is the peptide bond. Okay. Got it or not, all of you? Now this is the peptide bond. Okay. This is formed after losing this H and this H. Okay, H2O will be lost and it will be CO and NH. Okay, NH2 only. Okay, normally it is NH3, no? NH2 is what? Imino, isn't it? Imino in proline. Following all of you or not? Okay, so this bond is the peptide bond and there is no rotation possible about this bond. But rotation is possible about these bonds. Okay, about phi and psi bonds in an amino acid. Okay, and this is the rotation that allows it to fold into the alpha helix or the beta pleated structure. Okay, got it? Why is rotation prevented about the peptide bond? Because it has a partial double bond character. All of you who are going to appear for your professional examinations, these small tidbits that I tell you, they can be asked of you in your viva. So please focus, please pay attention, because anything that you read, you should know why, what, how, where. Okay, all these things, they can be asked in viva. Alright, so why is rotation prevented about the peptide bond? Because it has partial double bond character. Double bond is stronger. Okay. Now, what is this? Very easy. All of you would be knowing. It seems like a helix, so it will be an alpha helix. Okay. The phi angle in alpha helix, that is the angle between C and N, is minus 57 degrees. The psi angle, that is between C and carbonyl carbon, is minus 47 degrees. Okay. Now, what is the pitch? Pitch is the rise per turn. What is the meaning of rise per turn? What is pitch? Pitch, its definition can be asked, or in viva they can ask you. Pitch is the rise per turn. What is the meaning? It is the distance covered by the helix in one turn. Helix in one turn. In one turn of the helix, how much is the distance covered is called the pitch. Okay, so you should know, very very important. The examiner can ask you what do you call the rise per turn in an alpha helix. Okay, you during your viva, if they ask you something about, tell me about what is the psi angle, what is the phi angle, all these you'll be able to tell, all the fun do stuff. But when they come to basics, if they ask you what do you mean by, what do you uh, infer by the term rise per turn, okay, so this you should know. It is very basic. You should know that it is the pitch. What is the pitch? It is the distance covered by the alpha helix in one turn of the helix. Okay, from here to here, one turn. The yellow atoms are the carbon atoms and the blue atoms are the nitrogen atoms. Okay, got it? So this will be what? This will be the phi bond and this will be the psi bond, isn't it? Okay. Now the pitch here is 0.54 nanometers. Okay, that means one turn of the helix covers 0.54 nanometers. Okay, and how many residues are there in one turn? About 3.6 residues. Residues means amino acids. About 3.6 residues are there in one full turn. Okay, now later on in the next class we are going to read about the structure of collagen in detail, where we'll see how the structure of collagen, the pitch of collagen and the residues per turn in collagen are different from this basic alpha helix. Okay, understood? Now these alpha helices are stabilized by hydrogen bonds. Okay, hydrogen bonds. What are hydrogen bonds? They are the bonds between the oxygen of the carbonyl carbon in the peptide bond and the hydrogen of the peptide bond in the hydrogen present in the nitrogen of the peptide bond. See, this is the peptide bond, isn't it? This one was the peptide bond, as I told you. Now the oxygen in this hydrogen bond, the hydrogen bond has what? Bond between oxygen and hydrogen. Okay, oxygen is the acceptor, hydrogen is the donor. Now, here what happens is, this is the peptide bond. Okay, about this is the carbonyl carbon. Okay, the carbonyl carbon has an oxygen. Okay, so between this and between the hydrogen of this hydrogen present bonding with the nitrogen, between these two molecules there will be a hydrogen bond. Got it? I'll make it once more for it to be clear with you. Okay, suppose this is one amino acid. This is one amino acid. This is another amino acid. Okay, now these will form a peptide bond, isn't it? They will form a peptide bond. How? By the loss of water. Okay, so what will be left when they form a peptide bond? What will be left? Sorry, that was NH3 plus, no? NH3 plus. So NH2 will be left and COO will be left. CO will be left. Okay, got it? OH from here and H from here will be lost. Okay, now this H molecule along with this O molecule will form a hydrogen bond. Got it? This is the hydrogen bond. Oxygen of the peptide carbonyl and hydrogen of the peptide nitrogen of the fourth residue. Okay, got it? This is the hydrogen bond present in the alpha carbon. And these hydrogen bonds, provide the stability to the secondary structure. Okay. Now there are there is one important characteristic of the hydrogen bond, that when it is collinear, that is when it is occurring at 180 degree, then it is the most stable. Okay, but when there is some kind of a bend or an angle in the hydrogen bond, then it loses some of its stability. So any configuration of the molecule that provides 180 degree or collinear hydrogen bonds, that configuration will be the most stable. Okay, in alpha helix, the right-handed alpha helix is preferred, or right-handed alpha helix occurs in nature. Why? Because it has 180 degree hydrogen bonds. Okay, all right, very important. All right, now there is something called proline, which is an amino acid. Why? Because it contains RCH, COH. Okay, and what does it contain? NH2 rather than NH3 plus. So after this forms a peptide bond, it is not left with a hydrogen atom to contribute to the hydrogen bond. Okay, isn't it? After it has formed after this one has formed peptide bond, it is not left with any hydrogen to form a hydrogen bond. Okay, therefore proline can be incorporated only within the first turn of an alpha helix. Okay, and after that, because it does not have any hydrogen present on the amino group, therefore it cannot form hydrogen bonds in between, and therefore it cannot be incorporated in between the alpha helix. Therefore, whenever proline is present in between the alpha helix, it will lead to bends. Okay, it will lead to bend. Why? Because hydrogen bond is not present at that place. So there the structure will be weak. So it will bend at that point. Okay, so there is a question that has been asked time and again: which are the amino acids that induce loops or bends in the protein structure? The answer is glycine and proline. Proline, we have understood why, because it does not have hydrogen left after peptide bond formation to produce hydrogen bond. Okay, because it is an amino acid, it lacks one hydrogen. Okay, now what is wrong with glycine? Glycine is so small, it is so small that whenever it occurs, because it is the smallest amino acid. Okay, it does not have any R group. Okay, so what happens is, wherever it is present, it will go and induce a bend. Okay, because it is so small that it will go and fit itself inside the tight alpha helix structure. So this is the alpha helix configuration. Wherever glycine goes, at that place it fits very tightly and induces a loop or a bend. Okay, because of its small size. Okay, and this because it is an amino acid. Very important. Okay, so two amino acids that induce loops and bends are glycine and proline. Okay, now what are amphipathic helices? Can any of you tell me what are amphipathic helices? Amphipathic, we have discussed this term a lot of times. Amphipathic, what are amphipathic molecules? Amphi means both, right? Amphi means both. Pathy means space, isn't it? So amphipathic molecules are those that contain both hydrophobic as well as hydrophilic group, isn't it? Amphipathic, am I visible and audible? Yeah, great. So amphipathic molecules are those that contain both hydrophobic as well as hydrophilic groups, that is both non-polar as well as polar groups. Okay, now on one side of the alpha helix we have hydrophobic R groups, on the other side we have hydrophilic R groups. Okay, this is the alpha helix structure. On the R groups are arranged are present such that they point outwards. Okay. On one side we'll have hydrophobic R groups. On one side, on the other side we'll have hydrophilic R groups. Okay. What is the function of these amphipathic alpha helices? They will provide an interface between polar and non-polar regions. How? When they will occur wherever they will be present, this side, okay, if this side is hydrophobic, they'll make sure that the hydrophobic R groups point towards this side. The hydrophobic R groups point towards this side, and the hydrophilic R groups point outside. Okay, is this familiar? Is this structure familiar? Where have you seen this structure? In the cell membrane, isn't it? The phospholipids. The phospholipids are also amphipathic, isn't it? Remember how phospholipids are arranged? Phospholipid bilayer, isn't it? The hydrophobic hair, hydrophilic tail. Remember, and there are proteins arranged as pores or channels, isn't it? So these proteins, they have amphipathic helices. These carrier or channel proteins have amphipathic helices. Why? Because in the interior of the protein, there will be hydrophobic groups, and in the exterior there will be hydrophilic groups, and vice versa. Whatever, wherever they are placed, if suppose they are placed in a hydrophobic solution, the exterior will have hydrophobic groups and the interior will have hydrophilic groups. Okay, they will fold as such. If they are placed inside a hydrophilic solution, the exterior will have hydrophilic groups and the interior will have hydrophobic groups. Okay, this is the function of amphipathic helices. Okay, understood? Now these alpha helices are stabilized by the peptide chain. The primary structure was stabilized by the peptide bond. Okay, the peptide bond. The alpha helices, the main bonds that stabilize these helices are the hydrogen bonds between oxygen of one carbonyl carbon and the hydrogen of one nitrogen, isn't it? That is what we read on the fourth residue. These hydrogen bonds, they prefer right-handed alpha helix because there these hydrogen bonds are collinear. They don't have any bend. Okay. There are also van der Waals interactions present in the core of the helix, which tightly compact the helix. Okay, in that tightly compacted helix, the what interactions help the helix to be tightly compacted? The van der Waals interactions. Okay, so two types of bonds: hydrogen bonds, van der Waals interactions, and one more, the peptide bonds. Okay, peptide bonds will be present, won't they? Peptide bonds will be present between the amino acids, isn't it? They'll be carried from the primary structure. Got it, all of you? So we'll stop here and we'll resume in the next class with the beta pleated structure. Okay, this beta pleated sheet we'll resume in the next class that we'll start after a few minutes. Okay, all of you revise this, and do not forget to subscribe to this channel for more such videos, because these are classes that we are going to take in rapid succession for all of you, specially dedicated for all of you. Do share these classes amongst all of your friends so that you guys can sit and study together. Okay, and more of the doubt solving sessions can also take place. I'll keep in mind whatever you have mentioned in the chat here, and I'll make sure that I put up sessions that are beneficial for you before your professional examinations begin. Okay, all right. See you guys in a few minutes again with the next part of protein structure. Okay, bye bye.